large-scale MS and MSMS data sets. So Gary, are you here? Yes.
I am. Can you hear me?
So you can. Yep. Perfect.
Is the volume okay? Can you hear me fine?
We can hear you good. So
Okay.
You can share the screen.
Sometimes the computer takes a life of its own and dims my volume. And I will now, so I can share?
Yes. So we still go with the same rules, yeah? If you have questions, type them in the chat, don't be shy, and the speaker will answer some of your questions live immediately afterwards, and if we are out of time, the rest of the question can be answered in the chat.
So I trust you can see it now?
Yes, we do. Okay, go on.
Okay, very good. And this presentation, Maria, and thank you for the invitation, both to you and to Valerie for allowing me to talk a little bit about XCMS and also Metlin. This will have a foot in the door of a little bit of a tutorial, but also because there's been so many interesting and, at least to me, exciting things that have been taking place within our lab, I'm going to touch fairly heavily on some of the newer aspects of what we have that I'm sure you'll recognize immediately why I'm bringing them up and why they could be potentially very useful for this sort of research. I can break this talk down into three separate areas. One would be relevance. The second would be XCMS and an overview of XCMS. And the third is an integral part of XCMS is our Metlin technology, which is the database. So I usually end with the relevance, but in this particular case, I thought it would be interesting to start with relevance because it puts the following technologies into what I think is a nice perspective as to why we've developed them. So the relevance aspect of this is, again, we focus on mass spectrometry-based technologies. We have pretty much instrumentation from all the different vendors in our lab, Agilent, Bruker, Thermo, and Waters, and SciEx, and we develop these instruments for looking at particularly biological type problems. And before I start, I want to give special thanks to a couple of individuals who have really helped facilitate our efforts recently in the lipidomics field. Jean-Marie Galano has made some really nice contributions to our lab and also intellectually to some of the research that we're doing. And when I say in the contributions, not only from an intellectual perspective, but he's also sent some of his oxylipids to our lab for incorporation into the Metlin database. Also, Martin Gier has really gotten me well started into the lipidomics area, and again, we're now collaborating on many different levels. And also Cayman Chemical, who have been a really major contributor to making Metlin where it is today, as well as Avanti Polar Lipids. So back to the relevance. So relevance, what we typically try to do is we have these technologies, but we're also very interested in making sure that the technologies that we develop are useful for the individuals doing biology. So we're not simply developing the technologies for the sake of developing them. Where our ultimate hope is that we're creating things that can be useful across the community. And so the way we typically develop our technologies is that we have many active collaborations with biologists who are interested in identifying molecules that are associated with their particular systems. The microbiome has been an area of very high interest to us. And ultimately, the problems that we encounter when doing these sorts of experiments ultimately lead us to hopefully improve XCMS and also the Metlin approach, the database to facilitate this sort of research. So I really like to think of this as a synergistic type of relationship between ourselves and the biologists, and also having an online system as at XCMS Online and Metlin also being available online, ultimately leads to us getting a lot of feedback about how the technology is working and, more importantly, how it's not working. So we're constantly making changes to it. In the past week, we've probably made a number of roughly 5 or 6 different alterations as a result of feedback that we've gotten over the last couple of weeks. Again, those are relatively minor alterations. A couple of them were in basically structures that were found to be inaccurate in Metlin, and we just made the adjustments, and they've now been made immediately available in the correct form. So, I mean, obviously we put out this information in the form of publications, and we try to keep the keep our technologies moving in a forward type of direction. I think a series of papers that we've had have actually resulted in what I think is the most relevant aspect of what we're doing. And it ultimately results in what I think of as a very simple message, and a message that resonates with us, but also I think many of you out there, especially the biologists out there, this certainly should resonate with you as well. And that simple message is related to the central dogma of biology. So as we know, we go from DNA to RNA to proteins to metabolites and slash lipids in terms of the central dogma of biology. However, I think something that's really we've not appreciated, at least fully enough, is the fact that these lipids and metabolites interact with every level of the biology. As you know, for DNA signal transduction and epigenetic regulation, and RNA metabolism, metabolite sensing with riboswitches or post-transcriptional modifications, and proteins on many different levels, allosteric effects, catalysis by cofactors, and post-translational modifications. Of course, these types of interactions are well known to occur. And I think what I'm trying to point out is the fact that we now have tools in lipidomics and metabolomics that can allow us to more accurately identify these active metabolites or active lipids. So that's the primary point that I'm trying to make here. So the simple message in terms of relevance is that metabolomics and lipidomics, beyond identifying biomarkers and identifying the pathways associated with these molecules, is really a very powerful technology towards identifying metabolites that can modulate physiology. And if you're interested in this at all, there's many different examples in these 2 papers that highlight this reality. So now that's the relevant side of this. From a practical perspective, how have we been developing the technologies that allow us to do this? So XCMS is one of 2 principal technologies that we've been developing. XCMS and Metlin have been in the works now for some 20 years. So Metlin got started earlier. It was started in the early 2000s, so almost 20 years now since we've gotten started with the Metlin. XCMS was first developed in 2005, and so now for about 15 years we've been developing it. In 2012, we introduced it as the online system, and that's primarily what I'm going to describe to you right now, is the online development of XCMS. This is the interface that we have for XCMS, the XCMS Online system. It has obviously many different aspects to it that for analyzing your data. I think the simplest way to put this, and we actually have a large number of users right now. This number of 45,000 plus registered users is, I think there's definitely some redundancy in this number. I mean, I don't think there's that many metabolomics or lipidomics people out there that are actually doing mass spectrometry based metabolomics or lipidomics. But what I think is represented here is, one, the number of people who are interested in just using the Metlin database, for instance. So we have a large number of users who are not using XCMS, but they're primarily using the database. And we also have a lot of users who sign up multiple times for, and so there's a level of redundancy there in terms of them trying to increase their storage space. They create different accounts. And the other very big component to this large number of registered users is a reflection of this being a cloud-based system. So if you assume there's anywhere from 5,000 to 10,000 metabolomic/lipidomic individuals out there who are actually doing these types of experiments, they're typically working with a large number of collaborators who are equally interested in the data. So I think part of this 45,000 number is coming from people who are collaborating and have the data shared with them. So this is, and then in our lab, this is a very common process. So the individuals who are doing these sorts of, we as individuals who are doing these sorts of analyses will send the data that we generate through XCMS and just send them the links to that data, and then they have direct access to it. So it's a relatively straightforward process, and we believe on some level it's a fairly intuitive process for the collaborators, and they typically need a little instruction on how to use it, but in general it's fairly straightforward. Some of the interesting aspects of XCMS was that it was the original approach that nonlinear aligned the LCMS and GCMS data. So prior to this, the nonlinear alignment didn't exist for these sorts of separation-based approaches. And it turned out to be key in terms of correcting for the alterations that were taking place across the chromatogram. So this image that you see right here with respect to XCMS, that's the very first image that Colin Smith, who created the original nonlinear approach to correcting for the data. This image actually represents roughly a couple of hundred samples that were analyzed, and on the x-axis here, we have time. On the y-axis, we have the deviation of the chromatograms across the chromatogram. So these plots actually represent the correction that had occurred for each individual chromatogram. And as a result, you get something that looks much like this, where the data is now all aligned, and it allows for a much more accurate statistical assessment of the dysregulated peaks. So what Colin ultimately created was an approach that basically allowed you to first correct the chromatogram for basically misinformation. So basically allowing us not to compare peaks that were not related to each other, but more accurately compare peaks that were related to each other. And second, was the ability to do the statistical assessment of these individual molecules to pull out the molecules that were actually changing significantly with respect to p-values and also fold change, and ultimately pull out those molecules that would allow us to, in the next stage, identify them and hopefully gain some biological relevance with respect to those dysregulated molecules. We've also incorporated into XCMS some systems biology tools. This is largely the result of efforts on the part of Siuzdak Li out of Emory University. He's now at Jackson Labs based out of Connecticut. And basically it allows us to use metabolomic and lipidomic data to associate that data with pathway analysis. And we've also more recently added some cognitive analysis capabilities into XCMS, and with the ultimate goal of identifying active metabolites. So from a practical perspective, if you're interested in using XCMS Online, we typically give the users 25 gigabytes of space. However, if you find, like Jean-Marie recently found that he had a very large data set and he needed a larger piece of storage, he just contacted us and we bumped him up to 100 gigabytes. And that, I think in most cases, the 25 gigabytes is more than enough to handle any sort of data analysis. But if you do need additional space, feel free to contact us and we can bump you up. Another thing I think that's unique to XCMS or XCMS Online is support. I mean, a lot of the tools that are being created are oftentimes they're created and then they're just put out there and they're not taken care of in the long term. So we've actually now been taking care of XCMS Online for 10 years to make sure that the individual's experience is as positive as possible. I mean, of course, those of you out there who are using it on a regular basis, it does go down sometimes when we have system maintenance or issues at the Scripps Research in terms of the internet. But generally speaking, I think it's safe to say that 99% of the time the system is working quite well and quite consistently. And we also have many ongoing efforts to develop new technologies within XCMS. And perhaps one of the recent past ones that I like to talk about is one that's getting used more and more. And this was originally reported in Nature Methods in 2018, and we're continuing to develop this. And that's the fact that, as I'm sure you know, with XCMS Online, it's a technology that was developed for untargeted analyses. So, hopefully most of you are aware of what untargeted analyses are. Untargeted analyses are where you're looking at as many molecules as possible through these LCMS experiments, all within the same run. So it's not uncommon to see thousands of different types of peaks showing up, or features as we call them. And then ultimately the XCMS is designed, when you're comparing different types of samples, to ultimately drop that number down to a more manageable number in terms of the dysregulated peaks that point you toward the direction of which molecules are the most interesting with respect to the biological problem that you're interested in. But as many of you know, this is, and I think Maria kind of hinted at that in one of her slides, is that this is really the first stage in the whole process of understanding what's going on with respect to your samples. The untargeted analyses is then, at least in our lab, typically followed up with another set of untargeted analyses on a new biological set of molecules that have the same stresses in them, to help further validate what's taking place within these samples, and that we get and confirm that we're getting consistent results. The new component to XCMS has been what we call XCMS MRM, which is the next stage of validation for us, which is targeted sort of analyses with XCMS. And they do work in synergy, and I think what's especially nice about both XCMS Online and XCMS MRN is that they are platforms that are they're platform neutral technologies for analyzing both untargeted and targeted MS data. So when I say platform neutral, I mean that you can use, whether it's Waters triple quad instruments or Agilent triple quad instruments or Thermo triple quad instruments, it it doesn't matter, or if you want to use the Q exactive for the untargeted analyses, or QTOF from Agilent or Waters or Bruker or SciEx, XCMS Online and XCMS MRM will work for all these different types of data sets. And I just want to acknowledge two of the key individuals who were involved in the XCMS MRM. That was Xavi Domingo, who's now director of a lab in Spain, and Paul Benton, who is now at the Denali and a company based in San Francisco. All right, so I've given you a sense, hopefully, of the relevance and how XCMS is facilitating this sort of research. I think, and I'm sure many of you would agree, one of the most difficult and time-consuming parts of doing these sorts of analyses just begins when you finish doing the initial data analysis with XCMS or whatever platform that you're using. Because now you have to start, one, identifying what these molecules are, and number two, putting them into some sort of biological context that's meaningful with respect to the types of samples that you are analyzing. So Metlin has been, represents a huge amount of effort on our part to facilitate the identification process. And we're in the middle stages of integrating XCMS and Metlin more completely. So they are integrated somewhat, but the full integration to streamline this whole process is still ongoing. And I also would like to thank Cayman Chemical, especially from a lipid perspective, for helping facilitate some of the growth that we've seen in Metlin over the past couple of years, and also, as I mentioned before, Avante Polar Lipids. But Cayman has actually really facilitated, even in the early years, back in 2014 and 2015, they were primarily responsible for the early growth of Metlin. So basically what we're trying to do with respect to Metlin is we'd love it if you could see a molecule and within a few seconds identify it using MS-MS data, or get some level of preliminary identification using MS-MS data. However, that only occurs if the molecule exists in an MS-MS database. So this has been the major motivator of Metlin's growth using standards since 2003. Everything in Metlin is based on standards, and we've continued this idea over the course of the last roughly 18 years or so. Okay, so this is a slide that I'll explain to you why things took off so dramatically in roughly 2018 in a few minutes or so. But we clearly have been working very hard with Metlin for many years to try to increase its size. And in 2018, we had a real growth spurt, which is continuing today. We're still adding to the database, but currently we have 850,000 molecular standards in the database, each of which has experimental MS-MS data. And when I say experimental MS-MS data, I don't just mean we have one spectra on each molecule. What we have on each molecule, or what we've tried to obtain, I mean, in some cases, molecules don't ionize in negative ion mode, or some molecules don't ionize in positive ion mode. But what we've been trying to achieve with this has been generating positive and negative ionization data at multiple collision energies for each of these molecules. So each of these 850,000 molecules has at least 4 different collision energies, so data on it result from 4 different collision energies. And many of the molecules have multiple collision energy data in both positive and negative ionization mode. We test each of the molecules in both ionization modes to make sure it's right. So I think beyond its size, I think this level of systematic generation of data across multiple different collision energies and in positive and negative ionization mode really make METLAN quite special. And the reason why I think this is special is because oftentimes if you have MS data on a molecule, it can be very difficult to know if you just have this one-off spectra whether some of those peaks are noise or not. However, when you have multiple collision energies, you can see the peaks come and go as they grow in as you increase the collision energies. So it really adds another level of validation to what you're seeing is real. And I'll just add to this an ion mobility tease. There's obviously Jules gave a very nice presentation on ion mobility. We've actually been putting a lot of effort recently into increasing the CCS library of molecules that exist, and we're applying our automation technology now towards ion mobility CCS data. And that's something in the coming months we're going to be reporting on more extensively. But the growth is actually, at least in my mind, quite impressive. Okay, so METLAN statistics, we have roughly, right currently about 856,000 molecules. I think it's a little bit higher right now. Approximately 3 million molecules, or 3 million MS spectra. I mean, to me, the number counting spectra doesn't really mean very much. I think the primary thing that's meaningful is how many standards you actually have related, and how many molecules with actual experimental data that we have. And that's why I typically report how many standards we have for each for METLAN as it's been growing. As I mentioned, each of the molecules have been analyzed at 4 different collision energies, and they've also all been analyzed in both positive and negative ionization mode. That doesn't mean we have data in both modes for all molecules, because simply some molecules ionize better in one mode versus the other. And we have very strict filters in place that keep us from allowing data to go into METLAN that isn't of high quality. We really want to be very conscious of the fact that we don't want you to make these hopefully a false positive identification. And just to give you a sense of that, METLAN now has, in terms of these analyses that we performed, we have roughly a 74% success rate. And using our filters, we've roughly failed at 25% of the molecules. What this translates into is the fact that we've now analyzed over 1.15 million standards, and that's where ultimately the 850,000 molecules that we have in METLAN, that's why we report only 850,000, because we've only been successful and we've filtered out the remaining roughly 300,000 or so molecules, because the molecules didn't reach our expectations in terms of the data quality. And we have roughly 350 chemical classes. Anecdotally, I was looking at I was offered a data set recently, or a set of standards recently of 1,200 molecules, and these were human metabolites, or at least that's how they were described. So in order to make sure that we were making best use of our time, we went through the 1,200 that were being offered to us, and we found out that we were missing only 41 of those. So I think that's a nice testament to the comprehensiveness of the database and how effective it is now for helping identify molecules. For comparison purposes, this was published last year, so I'm sure there's been some growth in the database since then, but this just gives you a sense for where Metlin stands with respect to the other databases that have standard-based MSMS data. So obviously we're at a fairly nice size right now, but there really is tons of room to grow, and that's what we're trying to do right now. So I mentioned to you earlier what created this growth spurt that I mentioned, and the growth spurt was largely due to in 2017 we had this event in the lab. It was I was on vacation at the time, and I got this phone call from our security saying that something had happened in the lab, and we needed to I needed to address it. And I didn't know the extent of the problems that had occurred at the time, but later we saw the actual video when the 10,000 gallons of water came crashing down on our instrumentation, and ultimately disabled eight of our instruments quite significantly. So the reason I bring this up is that obviously this was a devastating experience. However, what we were able to do was as they were taking out some of the damaged equipment, we offered to purchase some of the instrumentation back. Obviously, fortunately, the insurance allowed us to replace most of the instruments, but we actually asked to purchase at a relatively low cost the damaged instruments, and of those, a couple of them we were able to resurrect, and they've become largely the heart of the growth of Metlin. So this is a real classic example of a bad year that we were able to turn into something, into a much bigger positive. So as I mentioned at the beginning, I mean, when we typically think of using MSMS data to identify molecules, you typically think of tandem mass spectrometry data. I mean, that's what how everybody now looks at, or what everybody uses to identify molecules and get confirmation of what the structure is. And that is defined by the fragmentation intensity versus M/Z. Here's an example of two molecules that Jean-Marie Galano provided to us. These are oxylipins, and you can see that they're very closely related oxylipins. I mean, the only difference is this keto version of the oxylipin of the phyto-P versus the hydroxy version. So they only differ by 2 mass units. However, when you look at the MSMS data, it really is quite striking. The fact that you only have one overlapping peak would strongly indicate that just by looking at the tandem mass spectrometry data, you'd never know that these molecules were as similar as they were. And this sort of an example, excuse me, got me thinking that we really need to develop Metlin in a completely different way to make it a more effective tool for identifying molecules, and especially in terms of similarity analysis. So what we've been doing, and actually we're doing this in multiple different ways, but I'm just going to describe to you one of those ways, which is the Metlin neutral loss database that we've created, which is instead of fragmentation intensity versus M/Z, now we can describe it as neutral loss intensity versus delta M/Z. So I'll go back to Jean-Marie's example, where we have the phyto-P versus the keto phyto-P. And when you look at the neutral loss mass spectrum, which is again a different way of thinking of tandem mass spectrometry data, which is again from a neutral loss perspective, and on the x-axis we have delta M/Z instead of M/Z, and the neutral loss intensity has been derived from the original intensities of the fragment ions. You can see that the the similarity is, in this particular case, much more striking. So the neutral loss data, and people have been using neutral loss data for decades. What we're doing here is just providing it in a different sort of way to help give us a better sense of where similarity lies between molecules, or if there exists similarity between molecules. So we're trying to use it to facilitate the identification process. Here's a couple of different examples. This is a genistein and methylgenistein molecule on the, on the left, you have the MS-MS data. On the right, you have the neutral loss data. Again, this is a nice example where there appears to be much more overlap in the neutral loss data than in the MS-MS data. Let's see. Actually, one of my favorite examples is the statin molecule. This is the rosuvastatin versus the demethylated version of that statin. And you can see here, this is the place where the methyl group is missing the methyl group. So these molecules are clearly very similar. The only difference in the structure is right here. However, the MS-MS data does not align in any sort of meaningful way. I think there's only 3 fragments that align between the two of them. However, when you look at the neutral loss data, it's actually much more, the alignment is much better. So again, I don't think the neutral loss by itself is going to be the best way of comparing different types of molecules. I think synergistically, the two different approaches for analyzing the data is going to be very effective. So I mentioned to you earlier the statistics for Metlin and gave you a sense of where the other database stood with respect to Metlin. And the neutral loss statistics are a little more striking. We have already created the Metlin neutral loss database from the 850,000 molecules that we have. And actually, surprisingly, there are other neutral loss databases of its kind that have been previously developed. So I think we have something here that I think is going to be fairly useful for the identification process, and it's we're actually facilitating its use right now in terms of things like doing developing scoring algorithms to further help identify not only the same molecules. Of course, those are easy because the MS-MS data should match exactly, but the molecules that are very similar to each other. And I'd like to acknowledge Aries A. Sporna and Winnie Heim for their help in developing, or actually their efforts in making this possible. So Maria, I'll just ask you, how much time do I have? I didn't really look when we first started.
I think you can go for another 5 minutes easily, and then we have a few questions.
Okay, perfect. That's about 5 minutes. That's about all I need for this very last portion, which I think is, to me, I've already told you about the relevance. I've told you about XCMS. I've told you about Metlin. I think this very last section is, I hope it gives you a sense of how we've tied them all together to create something that's kind of unique and also fairly useful from a broad global perspective. We call it ESA-MRM. What ESA, it refers to Enhanced In-Source Fragmentation Annotation. And ESA was developed in our lab as a result of something that hadn't been observed before. It had, and this was work that was developed, that Xavi Domingo made the original observation about this. So he was looking at in-source fragmentation, which we all know exists. We all know it happens. But Xavi's observation was that tandem mass spectrometry data that we had in Metlin matched very well with the in-source fragmentation data. And the reason why that's significant is the fact that you can now use instrumentation that doesn't have tandem mass spectrometry capabilities to generate data that is basically the same thing as when you're using a tandem mass spectrometer. So this is on the top here. This is data from from the fragmentation, from in-source fragmentation. Obviously, the intensities don't always match as well as this one does, but this is from the Metlin MS2 database. So this is the first part of the observation that actually we find is quite, at least to us, quite interesting. It allows people to do these sorts of identifications without having tandem mass spectrometry capabilities. But I don't think it's the most interesting aspect of this. I think the most interesting aspect of this is that when we use single quadrupole instruments in MRM mode, basically like you use a triple quadrupole mass spectrometer in multiple reaction monitoring mode, where you're just focusing in on a particular fragment, and in our case, a precursor as well, or a couple of different fragments, you can then get very good sensitivity, in some cases better sensitivity with the single quad than you would with the triple quad, simply because you don't have a collision cell where you get the collision cell losses. So this allows us to use single quadrupole instruments, much like you'd use a triple quadrupole mass spectrometer, with very high sensitivity, and ultimately allowing us to make use of an instrument that is 1/3 the cost for quantitative analysis and validation. And the reason why I mentioned that this is related to XCMS is because an algorithm that was developed with Martin Guerra and also with Rico Dirks, who is one of his staff in his lab, he used part of the XCMS technology to do alignment and also multi-sim sort of analyses to filter out peaks in these MRMs that weren't related to the molecule of interest. And so now we get ESA MRM data that looks a lot like what you'd see with the triple quad MRM transitions. And oftentimes the case, we're going to see them at higher intensities, and therefore presumably get the same level of sensitivity, if not higher. The other interesting thing about this is that our dynamic range with the ESA MRM approach is roughly 10 to the 5th or 10 to the 5. And we've done direct comparisons with triple quad MRM, and actually we see the same sort of dynamic range with both approaches. And we have multiple examples of that. There's a paper on this now in Chem Archives that you can look at, and hopefully it'll be published in a journal relatively soon. And with that, I think some of the implications of this is that, one, you can, as an alternative to using a triple quad analyses, you can use a single quad, but also it allows us to use a quadrupole time of flight mass spectrometers as devices that can do both targeted analysis with the untargeted analysis with the TOF, but also targeted quantitative analysis with the quadrupole. So you can actually have the whole package available to you for untargeted analyses and targeted analyses with the QTOF. And the individuals I'd like to acknowledge with respect to this has been Jingchuan Zhu and Martin Guerra, and as I mentioned, Rico Dirks, who developed the what we call correlated ion monitoring algorithm that allows us to do the filtering with this ESA approach. And actually, this is basically my last slide. I don't think I have time, so I'm not going to do the live demonstration unless somebody really thinks it's a good idea in the question period. But at this point, I'll be happy to answer any questions you might have.
Thank you very much for this very impressive presentation and very impressive work behind XCMS and Metlin. So many standards. This is really wow. MSMS spectra. We have quite a number of questions in the chart, but I guess majority of those are really practical. So maybe it will make more sense if you have time to go through them and reply directly for the application. So maybe we can ask live one or two questions here. For instance, there is a question for Whitney Stats. Whitney, would you like to ask your question?
Yes, hi Gary, thank you for the talk, and I really appreciate all the effort that have gone on to make Metlin happen, and I use it extensively for a lot of my work. But one of the struggles I have is figuring out how to download or export results so that I can then use those in report generation or to generate libraries for other software tools such as Compound Discoverer. So I was just wondering if you could comment on, you know, is there any way to submit a batch search and get something like an Excel file or CSV file?
Yeah, no, actually it's a very good question, and it's one that we get almost on a daily basis. So what I actually, multiple times I've submitted proposals for Metlin to get it funded so it would allow for that. And I think now roughly eight times we've been denied, unfortunately. So that's actually, that since we haven't been able to get funding for it, I have to come up with alternative sources for funding, which, and one of those alternative sources of funding has been to, now where it's being sold as a local version or a cloud version. So yeah, it's just basically a lack of funding that's keeping us from our ability to make it downloadable, because if we make it downloadable, basically we lose any chance of ever getting funding for it. So it's more of a practical consideration. But when you consider that this has been done without any funding, I think it's a nice thing to have online, but it does create some issues, I know, with individuals like yourself who want it to be downloadable. But it's maybe you can ask that question of the reviewers.
Yeah, I would love to see it be searchable in Compound Discoverer, so we could use that in our pipeline. And I don't know if vendors have reached out to you to have this as a possible function, but certainly actually would be valuable.
Yeah. So Waters does have a version of it in their system. I think they currently have the 100,000 version, and Bruker is, we're in a conversation with them to actually get that up and running for them as well. I do think I absolutely agree with you. I think it's much more valuable to have it directly associated with the program itself, and I think that's what the Waters users have found it to be valuable in that respect. And I think with eventually, once we have it fully integrated into XCMS, that'll be a valuable aspect of it as well. So we do have an API that's also available that would allow this to take place, but if you like, we can discuss that offline.
Great, thank you.
Sure.
There is a question from Joanna. Joanna, would you like to ask a question?
Yeah, thank you. First of all, great talk. I just want to comment that I fully agree with the usefulness of in-source fragmentation for annotation of metabolites, and I'm using this idea for annotation of metabolites for CE-TOF metabolomics for almost 10 years, and I was always very, very impressed how my in-source generated spectra and fragmentation spectra are similar to those from Metlin, and that was for me always a great source for information for annotation purposes. However, I observed some fragments that were generated for in-source during the in-source fragmentation that I couldn't see in the seed generated fragmentations. Did you observe such fragments?
No, actually, I think we've analyzed close to a couple of hundred now, and we've gotten really good consistency. But as with anything, I can imagine there's going to be exceptions to that rule. But generally speaking, the consistency has been quite good. It's really been, and I have to say the thing, of course, the identification part of this, and it always surprised me when Chavi came to me and we were discussing what his observations were, that the tandem mass spectrometry data matched the in-source fragmentation data so well, that nobody had ever published this before, because it was really quite surprising. And we even got that surprise from the reviewers when they were looking at the original paper. And and clearly you've made these observations for years, and the consistency, at least from what you just said, sounds like it's been generally quite good. To me, the most interesting thing about this, though, is not only is the identification very interesting, but the fact that you can use a single quadrupole in MRM mode to do quantitative analyses. I think basically this opens up roughly 100,000 mass spectrometers to doing quantitative analysis all over the world at a fraction of the cost. So I think if you ask me what part of this is most interesting, I agree that the in-source fragmentation matching the tandem mass spectrometry data is very interesting, but I think the fact that we can get equivalent sensitivity with a much simpler instrument is, to me, perhaps the most exciting part of this.
Thank you. And last question will be from Edward Dennis. Ed, would you like to ask a question?
Ed,
hi, Gary. Great talk from down the street.
Yes.
I'm curious if you could estimate of your 850,000 standards, what percent do you think are lipids? And then specifically human or mammalian lipids? Do you have any speculation?
God, to be honest, as you can imagine, without my primary focus has been to just get as many molecules as I can and to analyze them and develop the automation to do all of this. So, I mean, I guess I could refer to Jean-Marie Galano recently. Because we've provided him with the cloud version of Metlin, which actually he has a version that isn't the complete version, but it's got 600,000 molecules in it. And he said he was really pleasantly surprised at how many oxylipins were actually in there. But I can't give you a number. I can't even come close to giving you a number where, I mean, as I said, without getting funding for this, it really puts a strain on us to actually not only generate the data, but to analyze the data that we've been generating. So it's exciting, and please don't take this as a complaint, but that I wish we had more resources to look at this more closely. But I think in general,
everyone on the call shares that concern for more research money.
What's that?
Everyone on the call, I think, would like more research money.
Oh, no, actually, but Ed, I have to stop you there. We're talking about no money. There's a big difference there.
Okay, in the interest of time, we will move on now. Thank you very much, Gary, again. That was really wonderful presentation. There